IASTA. I am very happy to welcome you all to the 25th lecture in our monthly online lecture series, a special milestone for us. I am Manish Joshi, Secretary of Indian Aerosol Science and Technology Association. This association was founded back in 1988, and since then we have been bringing people together through conferences, schools, lectures to share knowledge on aerosol science and its applications. Under the leadership of Professor Sachinandan Tripathi from IIT Kanpur, our current executive committee started this monthly lecture series, and today we are proud to be hosting its 25th session. We are especially honored to have with us Professor Herrmann from the Leibniz Institute for Tropospherical Research, TROPOS, in Leipzig. He is a highly respected scientist in the field of aerosol research and also a professor of atmospheric chemistry at the University of Leipzig. Before we move on, just a quick reminder, the 14th Asian Aerosol Conference is coming up in Mumbai from 1st to 4th December 2025. Do check the conference website for details. If you haven't already submitted your abstract, please submit your abstract in a couple of days now. Abstract deadline is now approaching and 31st August is our final deadline. And if you have submitted your abstract, do not forget to register in time to take advantage of early bird deadline. Now coming back to today's lecture, which will cover wood burning, virus transmission, and microplastic atmospheric aerosol investigations, I would now like to hand it over to Dr. Abhijit Chatterjee, who is also our IASTA Executive Committee member, and who will now introduce our distinguished speaker, Professor Abhijit.
Thank you, Manish. Good evening, everyone. So I am Professor Abhijit Chatterjee from Bosch Institute, Kolkata, and I am one of the executive members of IASTA. So we are really honored to welcome Professor Herrmann, Professor of Atmospheric Chemistry at the University of Leipzig and head of the Atmospheric Chemistry Department at the Leibniz Institute for Tropospheric Research, TROPOS, in Leipzig. His research spanning nearly 4 decades has focused on atmospheric multi-phase processes with academic contributions at the universities of Göttingen, Hanover, and Essen in Germany, as well as the California Institute of Technology in USA. Since 1998, he has been based at the University of Leipzig and TROPOS, and has also served as concurrent professor at Fudan University in Shanghai, in China, since 2015. So Professor Herrmann is the recipient of numerous prestigious honors, including the President's International Fellowship Initiative, Distinguished Scientist Award from the Chinese Academy of Sciences, American Chemical Society's Division of Environmental Chemistry Honor Award, Environmental Science and Technology Reviewer Award, and the Gay-Lussac-Humboldt Prize as the first German atmospheric scientist to receive this distinction. He has taught atmospheric chemistry, earth sciences, and mineralogy at both the undergraduate and postgraduate levels at Leipzig University, Fudan University, and Shandong University since 2013. Professor Herrmann is a member of the Academia Europaea, the Leibniz Society of Sciences, and serves on the Evaluation Commission of the Czech Academy of Sciences, the Second International Indian Ocean Expedition, and several other high-level committees. He is also affiliated with renowned scientific societies, including CTAC, American Chemical Society, German Chemical Society, German Bunsen Society, and also he is a fellow of International Union of Pure and Applied Chemistry. With an impressive track record of supervising 56 PhD students and 57 postdocs, Professor Herrmann has authored 474 peer-reviewed journal articles, as well as 22 book chapters. According to Google Scholar, his work has been cited nearly 36,000 times and with an H-index of 95. So it is truly a privilege to have such an eminent scientist with us today. So please join me in welcoming Professor Herrmann. Professor Herrmann.
Thank you very much. Thank you. So I will first share the screen.
Yes, please.
And so I hope you see the title slide full screen. Thank you very much. I will start now. Thank you very much for the two friendly introductions. And it's true, we were just saying I started my first academic thesis work in 1986, so this is 39 years, so we are close to completing 4 decades of work. And also thank you very much for reminding me of some things in the extensive CV. But the nice thing about science, I mean, we want to always stay up to date. And so I hope I can present you a little bit like ongoing topics when we talk about atmospheric aerosols, which are interested in that would be the 3 mentioned ones: wood burning, virus transmission, and microplastics. And I'm pretty sure some of them are also of big interest for India. Yeah. And I did this with many co-workers, and not at least with Ankush Kaushik and Anju Peter for the micro and nanoplastics work, and I will come back to this later. But first, before we go to the science, I want to show you a little bit about Leipzig. Just one thing. This is our new university headquarters, and it looks a little bit maybe resembling like a Christian church. And this is true because there was a church actually part of the university headquarters for many centuries, but then it was taken down. And so this new building is resembling this a little bit. And so this is to advertise the city of Leipzig. We are located just a little bit south of Berlin. If you happen to be in Berlin, you can go here in 1 hour by train. Yeah, with a car it would be 2 hours. And then we have this building, which is maybe more pragmatic from its style. This is our chemistry laboratory building, or atmospheric chemistry department building, where we have our laboratories and where my whole group is working. And we are very happy that we have this just for the atmospheric chemistry. We moved into this in 2017. And this is what I want to do with you. I would like to walk you through the 3 chapters. Wood burning, maybe you are a little bit surprised, but this is a big topic for air quality in Germany. And then the second is virus transmission mediated through aerosol particles. And third, I come to micro and nanoplastics. So let's right away start with the wood burning. You know, this is of global interest because of climate change and the increase of wildfires. This is the one complex of problems which are caused by combustion. You see all the news that air quality in many regions of the world is dramatically deteriorated by the influence of wildfires. For example, if it's burning in Canada, it can even go down to New York City and cause a very bad air quality there. But it's a phenomenon that fire intensity and frequency is increasing a lot worldwide. And so this is this complex of wildfires or agricultural fires. This is also a big problem that farmers actually cleaned their fields by burning. In many countries, this is now not allowed anymore. In Germany, when I was very young, it was still allowed, but I would say it was forbidden already in the 1970s. In China, much more recently, and then different from this wood burning, we have wood burning for household heating. And this is maybe the surprise. I brought some pictures and newspapers. Here is in German, but you see here the picture on the left. many households have open fireplaces. This has this notion that this is very nice to have it in your living room and you have a wood fire in the evening. But this wood burning is one of the strongest sources of particulate matter in Germany. And since 2017, the PM emissions from this wood burning overran all the emissions from all the traffic sector in Germany. So we are doing something strange here. We are heating with wood and we are deteriorating the air quality in many regions. It could be countryside or also city parts, residential areas where we have really big problems. And sometimes people talk about the so-called wood stove gate, because they say, well, this is also a little bit triggered by politics, because for some time in the European Union, wood burning for household heating was rendered carbon neutral. And it was even subsidized when you installed such units and you said, I do my household heating, for example, with wood pellets. So it's an ongoing problem. And this table here, I'm sorry for the German, but this is the emission of certain things like PM10. You see here, this is the emission of PM10. And the unit is kilogram of PM10 per terajoule of energy. And you see here, gas is very clean. And the next column, this is wood in pieces. This is piece wood or log wood. And this column here is wood pellets. That is 1/3 of it, but we are in the area of 100 kilogram per terajoule per mole. And if you go to pellets, which have sometimes the image of much cleaner combustion, we are still in the area of 33 kilogram emission of PM per terajoule of energy, where natural gas has only 0.03 of these units. And so it's a big concern. Wood burning without after treatment is a big emission source. Yeah. And so our local environmental agency, we are in the local state of Saxony, approached us and said, could you really give us a quantification? How much can this wood burning contribute to very local air quality? This time for the countryside, where we know that many households and families are actually doing this. And maybe you have heard from literature that we have a research station. This is the Tropos research station Melpitz. And Melpitz indeed is a little village. And when I give this talk sometimes in China, it is hard to believe that there is villages like this where there's not 1 million people living, but in this village there's only 500 people. Yeah, and we have this in Germany, and I originate also from a very small city with 3,000 people. So we have this. And in this village, most houses have an open fire station, and we know this from the inventory. And so we said to the agency, okay, we will do our regular measurement at the research station, and just a little bit downwind in the main wind direction, we will measure also in the middle of this village. And this is a look from the station to the village, and you see a little bit smoke burning here. And then we went into this village, right into it, and put this measurement container there. This was very much insulated for noise, so it was quiet below 35 dBA, which is the rule, so you can hardly hear anything when it is operated and runs particle samplers and particle size distribution measurement, DMPS and things like this. And we put this in the middle of the Snellpits village, and we measured 1 year round, actually. And then this is from the proposal. We measured the ions, we measured carbon sum parameters, the anhydromonosaccharides like sugars for tracers for wood burning, the PAKs, alkanes, hopanes, and also metals. And then we tried to go after what we call the wood burning increment. And of course, we first started to do this gravimetric, and we just looked at the particle mass at our research station as a function of time in the year, throughout the year. And we see already directly quite a difference, like a little bit more than 1 microgram increase when we have a winter. And we were suspecting that this can have to do already with the wood burning. But of course, we did more analysis. We then derived how much wood burning contributed to the PM, and by the macro tracer method, let's say here with levoglucosan, we came to a number of between 2 and 3 microgram per cubic meter for the months of December, January, and February. Then we did a PMF analysis also, and we came up also with a little bit let's say with comparable but little bit lower numbers, like 1.5 microgram per cubic meter here. So when we look at the 3 approaches, gravimetric, macro tracer, and receptor modeling, we had results between 1 and 1.8 microgram per cubic meter. And we could say this is an increase of the combustion PM about 60%, which comes on top of the regional background. So the regional background also has PM from burning, but this is coming on a bigger scale. It is in the regional background, and the local increment is somewhere between 1 and 2 microgram. And so we said the best available value would be 1.2 microgram for PM10, and dependent on substances, we see much bigger differences. For example, benzoapyrene increased by 0.4 nanogram, which would be nearly 90%. And you might say, well, this is a very low mass contribution, but no. The limiting value for benzoapyrene in PM in the European Union is 1 nanogram. Yeah, and so it could be when you have just emissions from households doing this, that you come into a regime where you just have too much benzoapyrene in the particles, and this 1 nanogram per cubic meter limiting value will really become a problem. We published this, as was said in the introduction. I have strong links to the US, and I'm a member of the ACS, and I feel much accommodated since I was there many years ago at Caltech. And so I always recommend go there and be present. And so we went to ACS Environmental Go journal and published this. And also to make the risk a little bit understandable, we did a health risk analysis, and it turned out statistically the risk by this increase of these, let's say, a little bit more than 1 microgram per cubic meter biomass burning PM is comparable to the risk from the whole traffic if you are living in Germany. So the health risk for being injured or even die is comparable to participating in traffic. So this is written in this paper, maybe you would like to look it up. And then, as we often do, this is a field study, but now as a chemist interested in the molecular mechanisms, we also want to go to the laboratory and understand. And we always try to match this a little bit. So not just do the very descriptive field measurement, but also go after the processes. And for this, we go to a chamber. We call this the ACD, Atmospheric Chemistry Department chamber, ACD-C. And it is 18 cubic meter chamber. We have actually a twin chamber set up, and we can analyze everything and prepare particles. And we can also insert smoke. And the smoke can come from an oven, or it can also come from an open fire. We can also have like a barbecue grill where we just put some wood here, and either source can be connected to the chamber, and then we can do a photochemical aging of the smoke when we put it in the chamber. First, we empty the chamber and we clean it. And then we inject the smoke for a small period of time from the burning. We characterize it as good as possible. We look at the wall losses in this time already. And then at some point we turn the lamps on. We usually add another OH source, like hydrogen peroxide, to really get a decent level of hydroxyl radical. And then we can photochemical age the wood burning exhaust. And we can then have either continuous characterization by things like AMS running all the time, or we sample from time to time, or we just take a filter sample at the end, which would be after 4.5 hours, and we sample on filters for 30 minutes. And we had a student working on this, and we did these aging experiments for low NOx condition and high NOx condition. This is written here. And we also varied other parameters like relative humidity. And then the idea was, let's really look into, with targeted mass spectrometry, in the products which we see in these particles. And just, as you know, from wood burning, many mesoxyphenols are resulting, such as vanillin, for example. And this is very prominent. It had a very high single species concentration of 130 nanogram per cubic meter. And this would be beach burning before aging. And then if we age it for the 4.5 hours, we get this result. And this is to show you some things disappear out of the particles because they are converted. Yeah. So the chemistry is not finished if the emission is finished with the burning process and you get the smoke in the environment, but the atmosphere is aging the smoke plume and it is changing its composition. And this is why we have increases of compounds which are more functionalized, like the green one here, the 2,4-dinitrophenol, which is coming up. Yeah, so we get, by the photochemical processing, we get a change. Some compounds are reduced in their concentration and some compounds are increased. And we did this rather systematically for targets like this, and talking about increase or decrease. All these orange boxes indicate increases in concentration, and the blue one indicate decreasing concentration. And these matrices are for 3 different burning fuels like beech, birch, and wheat. We checked also wheat how does this behave? And you see here all the targets. Yeah. And so we have a big data set which we are currently investigating, and the topic of nitroaromatic compounds is of big interest. I know many groups working on this, and of course then we started to make a model module for our CAPRA mechanism. This is the chemical aqueous phase radical mechanism, and BBM would be biomass burning module 1. And so we started to degrade things like vanillin, syringol, guaiacol, and also levoglucosan, just these 4 compounds. And what you see here is levoglucosan, and I had up to now a postdoc and a master student working on this, and we are approaching already to 3,000 reactions, so it becomes quite complicated. If you look at this is a certain oxidation steps of levoglucosan where people have published that it can be degraded in the particle phase, but also in the gas phase. And if we look at this is just the start, and when you go to the bigger scheme, it becomes rather complicated. And you get a lot of functionalization and then decomposition. And we are working on this module, but it is far from complete, and it is getting very big, but at least we can do degradation of levoglucosan as a anhydrous sugar and 3-methoxyphenols. So this is something which we do in the mechanism development. Yeah, and then we can apply the model to campaigns. We did this for a campaign, for a measurement campaign over California, which has done by a consortium, I think led by NCAR, which is called WeCan, and the results are openly available, and we can get a hold on what the aqueous particles in the smoke plume do, and how the OH sinks and sources are if we have aqueous particles. And you see here that transition metal ion chemistry plays quite a big role over California. And in siphon, we see the most interesting thing only taking place when we have cloud activation. So the blue period here in the middle is cloud activation. And of course, this is one thing this CAPRA mechanism model was built for. So we can do some model calculations. However, the amount of organics we are treating is still quite limited. So I'm not reading this to you, but this is about the modeling. Maybe little announcement: if you are interested in modeling aerosol chemistry, I'm always saying contact us. This is open. You can get the mechanism, we can set it up for you so that you can run it. It comes in modules. If you type this into Google, you come to a landing page where you can get all these modules. And if somebody wants to use it, we do training. We do it sometimes in little classes that people come here, but we can also do it individually. And we will also organize a few schools for this. But if you are urgently in the position that you urgently want to do some particle phase chemistry modeling, just write to us, and it's very open, and we can help to get this going for you. It is used much in China, Europe, and in the US. And we are supporting this, so please contact us if you are interested. This is now bringing me to chapter number 2, so I think the 2 remaining chapters are a little bit shorter, not to talk for 2 hours here. The second thing is pathogens in aerosol particles, that is viruses. And what we wanted to do is we wanted to characterize how do aerosol particles in the chamber behave, which contain viruses, because you know from the pandemic there is not only the droplet infection, like the medical doctors learned, but we are, and you are all aerosol experts, we are exhaling population of aerosol particles into, say, indoors a room, where we have lifetimes of, could be hours of particles, which carry the infectious viruses. And so we were thinking maybe the chamber is a good thing to mimic this, but we need to build a good source to do this special exhaled virus, which contain a lot of material of organic gel-like substances, which also prevent fast evaporation. So this is something we were doing a lot, which took us quite some time for development. And Ali Nasrabadi worked with me. He's an aerosol specialist who worked before at Duisburg, which is very well known for aerosol physics and engineering. And he came to work on this virus project. And he developed this setup where we can atomize solution with the inorganic materials and then dry it a little bit. And then, let's say, modify the particles with these long ducts to grow more and all the time to contain, to keep the content of mucin, which is this gel material here. And then we did some degradation studies in our chamber, and different from many others who did not use the mucin, our particles are more realistic because they keep the humidity and they don't dry out so fast, which would be unrealistic if you leave this mucin away. And this is a look in our chamber laboratory. This is the conditioning, and this is a look below the chamber, and this is one of the 18 cubic meter chambers, and this is the biosampler. And we started to work with the T4 bacteriophage. We then continued a little bit until recently with this MS2 bacteriophage. And we got an allowance to do this, which is not easy, but we got, of course, no allowance to do the actually SARS-CoV-2 coronavirus because this is regarded too dangerous. So we have an S2 biological hazard level. And what we did then is we characterized the copies of a virus with a PCR, just like the doctors, the virologists did when they got samples from infected people. And so we just did the same from our samples, which we took with the biosamples. And first we looked at how linear this actually is, how many genome copies we have depending on the CT value of the PCR. And then we were able to change this with the, to check this with the actual setup for the virus in the chamber. And this was just a check where we expected nothing to happen for the two different viruses. They were just injected in the empty chamber with no light. And you see after 1 hour and after 2 hours, we could just quite well reproduce what was there. And then we could start to expose these virus populations in the dispersed particles to UVC. And you see here that depending on the UVC dose, which is given here in millijoule per square centimeter, the number of cycles in the PCR is increasing. And this means the number of virus or genome copies is decreasing. We have to cycle more in the PCR machine. And when we have like 400 millijoule per square centimeter for the T4 virus, there's nearly not any virus detectable anymore, only after 45 cycles, which is an awful lot. Yeah? And so this way we can quantify the effect of UVC on actually the population of the viruses. And we did this not only for UVC, but also for UVA. And even UVA is degrading the virus. You see it in this plot a little bit here. But you need higher energies, but then it is also possible. This is published. This is an early page of the of the paper which we submitted to Environment and Health, and we were happy that it's out, and we are writing now on more results, and we are trying to continue a little bit on this in a more applied setup, where we actually build this, which from the geometry resembles a German school classroom. They are very often like this. They have one side with windows. The area is like 35 square meters. They are like 3 meters high. And we equipped this with inlets for particles. You see it here, but also sampling. And you see here this torpedo-like tube thing. This is actually a UVC unit, which a company is selling. And below the ceiling here, this is another UVC unit, which also a company wants to test if these are feasible ways to come to virus-reduced breathing air indoors. Yeah, so it is a little bit like we call it pandemic preparedness. Sometimes we say we are pretty sure that there will be another pandemic coming. The question is only how bad will it be? And maybe we would be right to prepare for this a little bit. And luckily we had one project to set this up, and it will continue, but the funding for these things is not so as it was in the pandemic years. And also there's big political discussion if one should do things like this or not. Society, at least in Germany and in Europe, is a little bit skeptical if you come with such technical solution, let's say. Yeah. So, but we try to move on with this project. This classroom is like 2 or 3 of such installations which we have in Germany, so we can really do some studies under realistic conditions. So let's spend maybe 10 more minutes for chapter 3, some micro and nanoplastics. This is now where Ankush, who is in the audience, is working, and where we had Anju Peter working also both from India. So this is a little bit India in this science topic in my group. And they developed a very nice analytical method, and we started with polystyrene, and the method is based on Curie point pyrolysis, GCMS, and I have to admit that we are like lazy. We use the Curie point pyrolysis GCMS already 1990 when I started here in Leipzig, because it can do nice organics analysis with very low amount of absolute mass. So you can load Curie point foil with just one spot from a burner impactor with nanoparticles sampled, and you can determine alkanes or acids. Okay. Okay. Somebody maybe close the microphone. Okay, so we started with the polystyrene quantification, and then Ankush and co-workers did the quantification for this whole list of polymers like polystyrene we started with, and PVC, PET, and PMMA. And the nice thing is now we have a target list of for the quantification of these micro and nanoplastics in aerosol particles, which is about 15 target compounds. And Anju, Peter, and Ankush did a 14 days pre-campaign where we measured close to our science campus here and close to a street. And you see here for these days in September, the fine and the coarse mode particles. And that was our first feature, that we did this analysis from both PM2.5 and PM10, because this has hardly been done, and we more or less see like a 1-to-1 distribution between particles being 2.5 micron big or 10. Yeah, so like 50% of the microplastics are sitting in the coarse particles, and 50% in the fraction with sizes up to 2.5. And the masses which we see is about 0.3 microgram or 0.6 microgram in PM10. So we could say it is like a non-negligible fraction of organic mass or even mass of the PM in cities. And if you ask me what I think about it, my thinking is that it is really high time to assess this and to measure how much micro and nanoplastics we really have in the environmental aerosol particles. And I can tell you there's a big discussion of this in Germany, but there's probably areas in the world where this problem is much bigger. And then we can say the total results for the PM10 and the fine or coarse mode is like this. And here you see a lot of influence of car tire tread, which is from cars because we measured next to a street. And then we looked for sources, and I will skip this. This is like statistics. And Ankush also calculated possible health risk by this inhalation. And these numbers are about the first ones that we inhale something like 2 microgram when we look at the PM10 per day of inhalation, which is non-negligible. And we can then calculate health risk associated with this. And maybe the final remark now is, in each part today we used our chamber. And so, of course, we wanted to use the chamber also for the micro and nanoparticles, plastic particles, and we can try to introduce them into the dispersed aerosol. And we tried many methods, and we got good success with a vibrating shaker, so a combination of shaking and vibration, and to get this dispersed, because we wanted to check if the plastics are in particles, are they being degraded if we expose them to oxidants like ozone, UV, NO3 or OH, you see it here in blue, or is there nothing happening at all? And Anju Peter worked on this. The nice thing is we can apply the same analysis scheme like for the field measurements, the Curie point pyrolysis again, or we can use thermodesorption from cartridges and then GC MS. This is also possible, and she applied both. And you see here main result for a PS tracer, the polystyrene monomer. So if we did not do anything, we had the green bar. And then if we apply ozone plus UVA, it goes to the blue bar, and for the dimer the same and trimer the same. Or when we have toluene in the particles, you see the degradation here. And the yellow bar is after longer time, and the blue bar is after shorter time. So it looks like these things are being degraded when they are aerosol borne, and when they are exposed to oxidants or light in the atmosphere. And of course, we want to go after this now more. We see degradation of many compounds, and there have been also suggestions on mechanisms in the literature, like you see here. And the thing is now to search for degradation products. We can say we see some styrene from polystyrene, not so surprisingly. We see some toluene also. So there is a kind of fragmentation going on. But all of this is not working so effectively to the extent that the atmosphere is cleaning itself just after the emission of these plastic-containing particles. Question is, how big is this self-cleaning capacity? And this is currently ongoing work, and I think this could also be of broader importance. So let me summarize. Part one, I talked about wood burning. Maybe surprisingly, this is even a topic in Germany. Then part two is written here, aerosol knowledge on the virus transmission. It would be very helpful because this is at least a very important mechanism. And part three, micro and nanoplastics, it's like a beginning story that this is emerging to be important also for aerosol research. And so I think I wrote these clusters of aerosol chemistry research could also be of interest for India. And finally, I would like to thank you. I tried to write this here with the help of machine, but also with the help of Ankush. I hope it is just saying the same. And so I'm very grateful, Neeraj, for the invitation. And I hope it was interesting for you, and thank you very much for your kind attention.
Thank you. Thank you so much, Professor Herrmann. It was really very interesting and very informative talk. And there were all the three parts, very informative. So I am sure there will be multiple questions. So I would request people to raise hand, and then I will call them one by one. So I can see the raised hand by Rohit. Rohit?
Okay.
Yeah, Rohit, go ahead. Okay. So Priyanka? Yeah. Priyanka Agrawal. Please unmute yourself and then ask.
She's still muted, right?
Yeah, Miss. Okay, I saw the hand raised. Okay, I don't know, Miss. They raised the hand and probably it is down. So until they raise again, let me ask a few questions if you don't mind. So
Neeraj, you can just read the questions that has been posted to chat.
Yeah, Miss, that I will come at the end. So let me first, Miss, one-to-one, let me ask. So, Professor Herrmann, so I have a few, actually not questions, some clarification related to this micro and nanoplastics.
Yeah.
So you quantified them. So, Miss, can you run them directly, or first you have to do some kind of separation from other aerosols to avoid any kind of interference?
We use them in the matrix like we sampled them. And then this was treated with a solvent. There is a solvent treatment step after the sampling. So, sorry if this was not clear. usually the nice thing about Curie point pyrolysis is that we can just use the sample particle like from an impactor jet, and here is a pre-treatment with a solvent.
So that treatment will remove all other particles except plastic?
No, we are. No, the idea is not to separate them from the matrix, but the idea is to make the analysis work better. Actually, here, yeah. So there is still ions and things contained, but they are not interfering with this analysis.
Okay. Okay. Yes, Abhijit. Yeah.
Yeah, Professor Herrmann, thank you for your wonderful and engaging talk. So I have just a kind of very fundamental questions to you. Most of the nanoplastic or microplastics are insoluble in water. So my question is, how do the aqueous phase aerosol chemistry gets affected by the interaction between aerosols and the water insoluble microplastic? How does this aqueous phase aerosol chemistry get affected?
This is a very good question. And I have to say, for the aqueous phase, honestly, we did not go after this up to now. And I think because of this reason, yeah. However, if you have, so because these degradation studies in the chamber, that was not necessarily, that was not aqueous phase, that was just exposure of the aerosol to this oxidant. So that could have happened just with the plastic sitting at the surface of the particles. Yeah. And I personally, we have not started to do a dedicated aqueous phase chemistry with this. One could think about it, but I would be on your side and say they are not water soluble, so maybe aqueous chemistry should not be our first priority. Yeah, and the chemistry we see in the chamber is just the dispersed particles. Yeah, and it could just be surface chemistry. Yeah.
Right. Thank you.
Yeah,
Okay.
you're welcome.
Yeah. Okay. So other question by Ravi Singh, I can read from the chat box. So he's asking, can virus adsorb onto
Sorry, now there was a small interruption. I did not get the question.
Okay. Now am I audible?
Yes.
Okay. So the question was, can virus adsorb onto wood smoke derived aerosols? And does this affect their persistence in the air?
What can dissolve? Sorry.
Viruses adsorb onto wood smoke derived aerosols.
Yeah. So that this goes together, so to say. The two parts. And yes, of course. Yes, of course. Now we were mimicking the aerosol which contains the virus only with mucin solution. Yeah. But because we wanted to mimic the aerosol an infected person exhales. And we can do the same. We can make a wood-burning aerosol, and we can look at what does the virus do when it is taking up in such an aerosol. And we could look at what would be the concentration levels when this happens. Yeah. Up to now, we did not combine the viability of the viruses with using other kinds of aerosol particles, but this can be done. Yeah. it could be done.
Okay.
That would open a new venue that you say, okay, if I have this matrix of wood-burning aerosol particles, it is a good host for the viruses or not so good, you know?
Yeah.
And how does it change when you have oxidative chemistry going on? One could study this, yeah? That is a very good idea, but up to now we did not start this.
Oh.
But it's possible.
Okay. So another question by again Ravi Singh is, what sampling and analytical methods are most reliable for detecting microplastic in fine particulate fractions like PM2.5?
Which samplers are most reliable, you mean?
Sampling and analytical methods. Yeah. I think
Yeah.
this field is still in process. It is not yet established. I see different studies are doing different ways, but what are your thoughts on that?
Oh yeah. This should be very thoroughly studied. We are now using just standard high volume digital samplers, which have a flow rate of 1 cubic meter per minute and an inlet, you know. I have not seen particular criticism of doing this for microplastic. I have seen a lot of criticism for the microplastics analytics if it is doing in laboratories where you have additional plastic sources, and it starts with gloves. When people wear gloves in the laboratory, that they bring certain plastics into the samples. And so I saw concerns about this, but I did not see concerns about the kind of sampler. Yeah.
Okay. Another question is, can microplastic and soot particles act synergistically as carriers for viral pathogens and enhancing their long-range airborne transmission?
This is also good. That would be similar like wood burning particles. We could do soot particles and load them with plastic. And the question is, what would happen then, you know? Would the soot trigger some additional degradations? That could be. Or would it kind of inhibit degradation and makes the lifetimes bigger, and that would make the plastic survive for longer when you have long-range transport. Yeah, so it's the same thing. How does another matrix change the degradation, let's say kinetics or efficiency of the microplastic? This could be studied when we would dope soot aerosol with plastics and check for the lifetimes against non-soot containing aerosol. Yeah, that could be done also. So that's a lot of new studies.
Yes, it is a quite new field. Yeah.
Yes.
And another question is, okay. So can multi-phase chemistry of nanoparticles lead to unexpected response in PM concentration?
Aqueous chemistry of what?
Multi-phase chemistry of nanoparticles.
Very small.
Nanoplastics, actually. Yeah.
Yeah. It can be that the multi-phase chemistry is different. We have possibility to run our model in 64 size bins. And if you have things which are mainly in the nanometer size fractions of the aerosol, the chemistry can be different, yes. But to elucidate this and to isolate the effect, we would need to run that model in the bin mode, size resolved mode. It is possible, but numerically expensive. And we would need evidence that we have enrichment of certain compounds in the very small particles. If that is true, the chemistry will also be different.
Yes.
Yeah.
Okay. Okay. Another question by Ramya Sibalan is, I think she's want to ask related to the chamber study you were talking. So she is asking how well we could mimic the atmospheric condition in the chamber.
Oh, that's a good point. I would say quite well, quite well. We try to pay a lot of attention that, for example, OH we have in concentrations which we have outdoors, like plus minus 10 to the 6. Or NO3 radical, we go to 10 to the, like 5 times 10 to the 8 per cubic centimeter, I'm sorry. And we don't overdo the oxidation levels. We have we take a few ppb of ozone, like 10 ppb or something like this. And we don't run at extreme oxidation levels like in an OFR, like an oxidation flow reactor, where you try to accelerate atmospheric oxidation. So we try to be as close as possible to the environmental atmospheric concentration, also for NOx, but especially the oxidants.
Okay. And the last question which I see is by again a PhD student Swati Joshi from Delhi University, and she is asking, can you share insights on which NACs dominate under biomass burning condition? Also, in your, yeah, can you hear me?
Yeah, can you repeat?
Yes.
Yeah.
So she's asking, can you share insights on which NACs dominate under biomass conditions?
Which NACs? Yeah. It is about those which I was shortly showing. It is things like 2,4-dinitrophenol is very prominent, but they can also all the methoxyphenols. They can also be nitrated. Yeah. So if we have 2,4-methoxyphenol, it could also be nitrated. So all of these combinations can be found. And at the end, you get some substances like the one I mentioned here, 2,4-dinitrophenol, and we have nitrophenol, nitroguacol, nitrocatechol, methylcatechol. So the most important NACs is probably those down here, yeah. From 3-methyl-4-nitrophenol, 2,6-dinitro-4-methylphenol, 2,4-dinitrophenol, which I mentioned, 2-nitrophenol. Even though it escapes to the gas phase, I would add 4-nitrophenol, like up here, 5-nitroguacol, very low, and then final one, 2-methyl-4-nitrophenol. Yeah. So that this maybe you can do a snapshot from the slide. This is the most important NACs which we see.
Okay. Okay, and then another question which you probably already answered. So is Anil Singh Salai, as you can see, he was asking again that how do you carry out the sampling, but he has already answered that.
Okay.
Okay, and okay, I can see one more hand is. I hope this it is okay. Can we take a few more questions?
Yeah, of course.
Okay, Indra Mohan, yeah, please go ahead.
Hello, sir. I am Indra Mohan from IIT Kanpur. So I have a question. Like here in India, during the COVID outbreak, the number of cases in winters were higher as compared to summer season. So is it anything related to like the liquid water chemistry or anything like that?
Neeraj, can you help me?
Okay, yes. Okay, let me repeat his question. So he's saying that during COVID times, during winter, the virus were more compared to summer. So he's asking whether anything atmospheric chemistry was causing this thing. Yeah.
Yeah, this is a very good question. And it could be that we just have oxidative chemistry. We have OH formation in summer. And of course, the OH is attacking the virus also. Yeah. And so it would simply be more oxidative force attacking the virus, which is in the aerosol particles, which would fight for us a little bit. And if we have winter conditions, this is breaking away and gives longer virus survival or bigger survival rates for the virus.
But sir, the oxidation will eventually kill the virus, not increase it. The oxidation will deplete the virus, right? The membranes of virus, or it will be destroyed by it.
Yeah. I did not report this. We studied this. We did not study only UVA and UVC, but we studied also production of OH by hydrogen peroxide, so that we mimic this, and we saw also a big reduction in viability of the virus. Yeah,
Okay.
and that would be what we expect in summer also. Yeah,
Okay.
very good question.
Okay, because the summers and winters here in India are just opposite or very different from that there in Germany.
Oh yeah.
Yes, because we have seen that because I just attended the OPTEC training in the April, so I have seen that difference.
Okay.
Okay, yes, sir.
Okay. ठीक है.
Yes.
Yeah. So, with this, so we are end to the question answer session. And now I request our colleague, Dr. Amit Kumar, for the vote of thanks. Amit?
Yes, sir. So now we came last part of the lecture. So a very good evening, everyone, and thank you for staying with us till the end. On behalf of IASTA, I have the pleasure to thanking our speaker, Professor Herrmann, for his outstanding and highly topical lecture on wood burning, virus transmission, and microplastic. So he has gave us valuable scientific insight and many ideas to take back to our lab and modeling work. So my thank also to Dr. Manish Joshi, Secretary of IASTA, for starting the session, to Dr. Abhijit Chatterjee, EC member of IASTA, for introduction of the speaker, and Dr. Neeraj Rastogi, Vice President of IASTA, for his managing excellence question answer and smooth moderation, which made the event run on time. We are grateful to Professor Herrmann for taking his time to join from the Lepis to our speaker and to all participants from their thoughtful question. Thank you everyone for IASTA and sponsor for enabling multi-lecture series. And I request everyone please join us for the next IASTA monthly lecture, and also remember to submit Asian Aerosol conference paper. The last date of conference paper is 31st August 2025. Thank you once again, Professor Herrmann, and thank you all. Jai Hind.
Thank you. Thank you, Professor Herrmann. Yeah.
Thank you very much. Thank you.
Thank you.
Bye-bye.
Bye-bye.
Bye-bye.